Good morning. It's May 19th, and this is your daily brief in science. Here's everything you need to know. A new Phase 3 result puts sacituzumab tirumotecan, or sac-TMT, at the center of endometrial cancer treatment after platinum and PD-1/PD-L1 therapy, showing gains in overall survival and progression-free survival versus physician's choice chemotherapy. Interim data from TroFuse-005 indicates statistically significant and clinically meaningful improvements in OS and PFS when sac-TMT is compared with doxorubicin or paclitaxel, and the study met its key secondary endpoint of objective response rate. Merck and Kelun Biotech report that primary endpoints were met and that survival improved versus physician's choice chemotherapy, with safety signals aligning with prior studies and no new concerns. Data will be shared with regulatory authorities worldwide as Merck advances approvals and global commercialization potential. The sac-TMT program follows earlier TROP2-directed ADCs on the market, highlighting competitive dynamics with Trodelvy and Datroway in the field. Merck is expanding sac-TMT as Keytruda's exclusivity wanes, aiming to strengthen its oncology portfolio amid evolving revenue expectations. A Commissioner's National Priority Voucher was awarded to accelerate FDA review of sac-TMT, potentially shortening development timelines. TroFuse-005 results were developed by Merck Research Laboratories and announced in May 2026, with additional data already presented at prior conferences. Dr. Dean Lee described the TroFuse program's aim to maximize tumor payload delivery while limiting impact on healthy tissue. The collaboration supports a broad sac-TMT program with 17 ongoing global Phase 3 studies, with results from lung and breast cancer trials anticipated at ASCO. The TroFuse program, exclusive to Merck outside Greater China, seeks to evaluate sac-TMT across multiple cancers, including endometrial, bladder, breast, cervical, gastric, non-small cell lung cancer, and ovarian cancers, in various settings and combinations with immunotherapies. A new study uses math, simulations, and neural networks to show how recognition and memory can promote cooperation even in simple settings. In particular, neural networks simulate repeated prisoner's dilemma games to test opponent-specific reactions and how they foster cooperative behavior over time. Across fields, from mathematics to statistical mechanics to neural networks, the work finds that cooperation can arise without external enforcement or kinship, suggesting stable patterns can emerge in simple interaction rules. A core theoretical advance accompanies these results, a generalization of Fisher's fundamental theorem of natural selection in evolutionary dynamics, presented as a generalized Fisher theorem within the study. Led by Alexandra Morozov and Alexander Feigl and published in the Proceedings of the National Academy of Sciences, the Rutgers-led work links cognitive-like recognition to emergent cooperative behavior and argues that cooperation can arise naturally in the prisoner's dilemma without extra rules, genetic ties, or kinship. The framework extends to multi-system cooperation, indicating periods of stability and upheaval over time, and raising implications for understanding cooperation in human societies and other complex systems. HistoSonics is pushing deeper into Asia, using TFDA approval to speed adoption, expand clinical evidence, and grow training across the region. The company is pursuing de novo FDA clearance to extend the Edison system to kidney tumors, backed by the HOPE4KIDNEY trial, which enrolled 67 patients and wrapped enrollment in mid-2025, underscoring nephron-sparing potential. The TFDA granted approval for the Edison histotrypsy system in Taiwan, marking a milestone for Asia-focused growth and global expansion. National Taiwan University Hospital has treated liver, kidney, pancreatic cancers, and sarcoma under research protocols, signaling broad early clinical exploration of histotrypsy. Histotrypsy uses mechanical cavitation from focused ultrasound to liquefy targeted tissue without heat, suggesting potential for outpatient treatment and rapid recovery. NTUH's clinical leadership is a central driver of HistoSonics' regional strategy and future applications. In the U.S., Edison has de novo clearance since 2023 for liver tumors, making it the first histotrypsy platform in the country, with about 90% tumor control in one-year data. TFDA's review process was thorough and ended in unanimous committee support. CEO Mike Blue calls the TFDA approval a validation of strong clinical evidence, regulatory rigor, and histotripsy's potential as a non-invasive therapy platform. NTUH was the first installation site in Taiwan, where physicians quickly adopted histotripsy under research protocols, reflecting rapid clinical uptake. A long-term Swedish study is tracking children from infancy to age 10 to map early signs of autism and ADHD using a wide set of methods. Early Autism/ADHD Sweden, or EASE, combines eye tracking, EEG, MRI, behavioral assessments, and gut microbiota analysis to chart brain development over time. The project is a collaboration between Karolinska Institutet's Kind Center and Uppsala University's Dive Lab, applying measures from infancy onward to understand typical development and early deviations within the same framework. Participants include children with and without close relatives affected by autism or ADHD, enabling direct comparison across development within a shared study design. Researchers aim to identify potential autism subgroups, such as those with later emerging symptoms, and to understand factors shaping long-term developmental trajectories. A core goal is to assess the stability of early diagnoses over time and explore whether subgroups exist among autism cases. Early findings show infants later diagnosed with autism often follow others' gaze similarly, but have reduced initiation of joint attention by 10 months, suggesting early social motor differences rather than perceptual deficits. 3-year-olds with autism may show heightened ability to detect visual details in complex images, indicating variability in cognitive processing. Infants who later receive an autism diagnosis may exhibit stronger pupil reactions to changes in light and to non-social sounds, pointing to early differences in attention and sensory processing. The extension will support investigation into whether infancy measures, brain activity, eye movements, and behavior predict later development, and will foster international collaborations on ADHD trajectories. A SEK 10 million grant from the Marianne and Marcus Wallenberg Foundation extends the study to age 10, enabling analysis of diagnosis stability, developmental trajectories, and the development of AI-based tools to predict diagnoses at age 10 from data collected at age 3. The extension also supports evaluating whether infancy measures correlate with later outcomes and will enable AI-driven prediction tools using early data. The 5-year donation from the Wallenberg Foundation funds the extension to 10 years, focusing on trajectory mapping and stability of early diagnoses. A new multi-organ-on-a-chip project called GlucoBrain aims to map the diabetes to dementia connection by linking gut, pancreas, and brain in a single 3D microfluidic platform. Researchers start by modeling each organ separately, then connect them to study glucose regulation, neuronal viability, and gut hormone signaling with bidirectional feedback loops. The goal is faster, more predictive drug discovery, with less animal testing and AI analysis of multi-parameter data to track disease progression and personalized treatment. The platform uses living human cells in advanced lab-on-chip systems to recreate tissue structures and provide physiologically relevant nutrient delivery, gradients, and mechanical cues for inter-organ signaling research. Funding from the EPSRC Health Technologies Connectivity Awards underlines a collaborative, interdisciplinary approach to tackling diabetes-related cognitive decline and therapies. A core aim is to modulate glucose concentrations, hormone gradients, and drug interventions on the chip to observe how diabetic metabolic stress affects neuronal function and cognition. Key questions include how gut and pancreas signals influence brain health, why organ-on-chip models may outperform animal testing, and how patient-specific chips could guide treatment choices. The device will be a multi-organ biochip enabling real-time tracking of signaling and cellular responses to changing glucose and hormone levels, allowing direct observation of inter-organ communication. Dr. Moshu emphasizes real-time observation and manipulation of gut-pancreas-brain chemical conversations as essential to addressing root causes of diabetes-associated cognitive decline. The project seeks to broaden understanding of diabetes and dementia, potentially yielding new treatments and more human-relevant testing platforms in medicine. Beyond basic science, GlucoBrain aims to validate drug behaviors on human cells, accelerate discovery, reduce animal testing, and pave the way for personalized medicine using patient-derived cells. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.